Both clams and mussels belong in phylum Mollusca and class Bivalvia. Both species are filter feeders found in marine and freshwater habitat. While marine mussels are most abundant in low and mid-intertidal areas in temperate seas worldwide, some species live in tropical intertidal zones and even salt marshes. Marine mussels are often found clumping together on rocks. One reason they live in groups is to keep moisture in mussel bed to prevent drying out while exposed to air during low tides. Marine clams, on the other hand, are commonly found in coastal waters. They like to burrow about 1 to 2 inches into sandy bottoms. And similar to mussels, clams are often found in groups.
Like many other bivalves, the anatomy of clams and mussels are one and the same. Since clams and mussels are soft-bodied animals, they possess a hard exterior shell that helps protect them from predators. The shells of clams and mussels are varied by size, thickness, and coloration, depending on what type of species they are within their clam and mussel family. Rings surrounding the shell of the animal are a good determiner of the animal's age. They use the anterior and posterior abductor muscles to open and close their shells. Within the interior lies the heart, kidney, anus, siphon, gills, intestines, reproductive organs, foot, mouth, stomach, and digestive gland.
Even though both species belong to class Bivalvia, clams and mussels acquire different habits. Mussels are the most motile among bivalvias. They have an oval or circular shape, but clams are only oval-shaped. Moreover, while mussels are found adapted in saltwater and freshwater in a range of conditions, mussels are rather adaptable to fluctuating environments because of its ability to survive while exposed to air.
Reproduction for both clams and mussels occurs sexually. Fertilization occurs when the eggs and sperm are released into open water. Once fertilized, the developmental growth of clams and mussels occurs in 3 stages. The first stage in development is known as the trochophore stage. Trochophore larvae are spherical and pear-like in shape and is girdled by a ring of cilia. During this stage, the trochophore larvae feeds entirely on plankton for survival. In the second stage of development, the trochophore larvae goes through 2 transitions: early development, known as veliger, and late development, known as pediveliger. In the first transition, the veliger larvae starts to form a foot and shell. This formation of the foot leads to the locomotion of the larvae that is seen in the pediveliger stage. To reach the juvenile stage of development, metamorphosis of the larvae needs to occur. Metamorphism is induced by chemical cues. These cues help the juvenile begin the shell calcification process, which is needed for protection and survival. During metamorphosis, the larvae undergoes internal and external changes present during the second stage of development. Once the juvenile stage is reached, the clams and mussels are pretty much formed. Clams and mussels are then considered adults once they reach sexual maturity.
One of the mussel's biggest predators besides humans are the sea star. The sea star slowly finds its way towards its mussel prey using tiny sensory tube feet, which will be used as suction cups to pry open the mussel's shell. When the mussel figures out it's about to be lunched, the abductor muscles will then clamp down against the sea star's tube feet. Once the sea star outmuscles the mussel, it ejects its translucent stomach into the mussel's shell and begins to unfold. The stomach then spews digestive chemicals that dissolve the mussel's flesh down to a liquid, which is then absorbed by the sea star. Once finished with its meal, the sea star then retracts its stomach and leaves behind a slick and shiny shell. Just like the mussel, sea stars also enjoy eating clams, as do other fishes, crabs, snails, marine mammals, and shorebirds. Besides the basic battle of sea star versus shellfish, marine mammals and shorebirds also have learned to outmuscle the clam. Although instead of using suction cups to pop open the shellfish, marine mammals like the sea otter bang the shells against rocks and boulders to crack open the calcified shell to devour the soft pink meat. Seabirds use the same strategy by raising the clam and dropping it on boulders below to crack the clam's shell. Snails, like the deadly cone snail, drill a perfectly circular hole into the top of the shell and suck their prey dry. Clams have to work hard to prevent getting eaten, but on the contrast, don't have to work as hard to eat. Clams contain 2 siphons which allow the clam to bring in water towards its gills. The gills not only are used to collect oxygen from the water, but also contain cilia which filter out small organisms called plankton. Mussels use the same feeding techniques as clams do, by siphoning water in towards the gills while cilia on the gills catch plankton, bacteria, decomposing plants, and animals. The gills secrete a net which trap the filtered plankton and then move the captured food over the palps and towards the mouth where digestion takes place. While learning about the nature of clams and mussels in their natural habitat, it is important to consider the impacts humans have on the marine ecosystem. Marine pollution such as oil spills, plastic buildup, and carbon dioxide damage marine habitat and those who inhabit it. Therefore, we as humans should be more aware of the hazards we cause to our environment.